Hi, I'm Nathan Lee,
I'm Alan Lowe.
I'm Gavin Seiden.
and we've done a thing on sugar-free sweeteners and the microbiome. No, it's this. Okay, so sugar is found in a lot of things that we eat, like desserts, candies, and drinks. But sugar is not very good for you because it causes blood sugar increases, which can lead to obesity, diabetes, fatty liver, and other bad things. So a lot of people will choose for like a light or like sugar-free alternative. And then the problem is that these artificial sweeteners usually have like side effects that affect your gastrointestinal microbiome. For example, maltitol, found in like sugar-free gummy bears, causes stomach pain and gas. So our study is going to find a solution to the side effects of like maltitol. All right, so the microbiome, before you have sugar-free sweeteners in the stomach, has a lot of bacteria, mainly from 5 phyla, Firmicutes, Actinobacteria, Bacteroidetes, Fusobacteria, and Proteobacteria. For example, lactobacilli converts nitrates into nitrites, and it helps prevent like pathogenic yeast growth and eczema. It's like it makes cytokines to inhibit skin inflammation and bacteriocins to kill other bacteria. So that's like supposed to be there. It's good for you. And then in your intestine, you've got thousands of bacteria, mainly from 2 genera, like the one on the left, the one on the right. And then the ratio of like the bacteria you have is caused by like your diet. For example, if you eat a lot of whole grains, you're going to have more Prevotella because they help you break down fiber and starch. Another type of bacteria is Bifidobacteria. You find more of it in breastfed babies because it helps them break down the human milk oligosaccharides and helps them make stronger gut mucus. And they live in the stomach, gastric mucosa, and the intestines. They live on the outer layer of mucus on top of the denser one that's on top of the epithelium.
Okay, so based on a study we found that was conducted on mice, the researchers were able to determine that levels of Bifidobacteria, lactobacilli, and SCFAs, or short-chain fatty acids, were increased in mice that replaced their diet from sucrose to sucrose being natural sugar, and they replaced the sucrose with maltitol. And the effect on the microbiome was generally good because fecal bifidobacteria is very common. It's very beneficial. It protects your gut from infections. Lactobacilli is also very common, and it's used in a lot of probiotics. Although it's important to note that having too much of any one thing in your system can be bad for you. So there was also heightened levels of SCFAs, or short-chain fatty acids, and which is pretty much the only truly worrisome result because SCFAs may decrease risk of infection, parasites, and cancer, but they also generally contribute to diabetes and obesity, which are 2 very common ailments today. So this may show that the problem may not originate solely within the microbiome or changes to the microbiome.
In a study done in Germany in 1990, researchers radioactively tagged maltitol and fed it to both conventional and germ-free mice. In germ-free mice, in the excrement of germ-free mice, there was 39% radioactivity, and there was 59% radioactivity in the air. In conventional mice, the excrement contained 20% radioactivity and 72 to 74% in the air. The germs account for the 19% difference in excrement and the difference in the air as well. The takeaway is that the germs assist in the digestion of maltitol. The following are not related to the aforementioned study. Maltitol is broken down by bacteria using the enzyme maltase, hydrolyzed into maltose and sorbitol. The maltose is broken down by a bacteria into glucose, which is digested into carbon dioxide and water, which leaves through excrement. And sorbitol is also digested, which also produces carbon dioxide and water. For each mole of maltose, up to 12 moles of water and 12 moles of carbon dioxide is retrieved, and the water causes the diarrhea, and CO2 causes gas.
All right, so what we plan to study is, first of all, we're gonna take the most common gut bacteria. We're gonna put them on a petri dish. Then we're gonna expose them to maltitol, and then we're gonna monitor the amount of CO2 they produce, because that will show us how much of the maltitol is being digested. And then we'll take the bacteria that do the most digestion, and then we'll attach fluorescent proteins to their maltase enzymes, and then we'll give them maltitol that is radioactively tagged with carbon-11, and then we're going to use fluorescence microscopy, STED microscopy, to watch as the maltitol enters the bacteria and then gets broken down, and then ultimately we're going to try and figure out whether we should prevent maltitol from entering the bacteria, or if we could just disable maltase, because you don't have to digest something to taste its sweetness. It can just pass through you undigested, and then you wouldn't have any side effects. So that's our goal. So you can eat sugar-free gummy bears without having diarrhea. And these are our references. Okay, questions? There is no slide after this. Just stretching. Okay.
Can you guys, so the last slide, can you go back?
Oh.
We just dig a little bit into what you plan to do, right? So you went through that. There's a number of different techniques, right? So what exactly what's the sequence again? Can you go through that again? And then let us sort of describe in greater detail when you're looking at these bacteria, right, that have maltitol in their environment. What do you hope to see from the optical microscopy that you're utilizing?
We want to find out we want to find out first which bacteria are mostly responsible for maltitol being digested, because there's thousands of them. You can't test all of them. And then so with the fluorescence microscopy, we're gonna attach fluorescent proteins to their maltase enzymes, and then it's kind of complicated, but we were hoping to use carbon-11 to attach to the maltitol, and then use positron emission tomography to watch as that would go inside the bacterial cell, and then we could see with the STD microscopy it being broken down. So then we could figure out a way to stop maltitol from going through the membrane, so then it would never get digested at all. Okay, epic.
I'm just thinking about the complexities of that experiment.
So I think that's it's kind of ambitious to think. I think one thing you have to consider is radio labeling does not have any kind of high resolution, right? So labeling will give you total gross measurements from the whole dish, right?
Oh.
But what you're doing is actually possible. It's tricky, right? But I'll give you two extra techniques. We haven't actually covered it this week, but it's a good opportunity to bring this up. You have one more bit of what we can do with light imaging technology. Is, for example, in that whole field of diverse bacteria, you have a challenge. You wanted to identify the ones that are actually doing the most metabolism, for example, right? You want to identify the ones that are uptaking the product the most. So maybe if you had, labeled maltose or labeled byproducts that you could look at optically and identify which ones were consuming the most, you could then actually cut those bacteria out, and you could sequence just those bacteria. That would allow you to identify exactly which bacteria were the culprits from a big mix of them. right? And that is something we do, is laser microdissection. You can actually use a laser to cut out just that one square where you think the offenders are, right? I think you have to think a little bit more about how you make the signal of who's producing the most maltose. Maybe you need the byproduct of maltose digestion to be fluorescent. So those bacteria that are making the most of those short fatty acids are the ones that light up. Things like that, right? So there's a couple practical details. But other than that, it is possible to accomplish what you want in there. And then if you want to use an optical method to monitor the enzymes and look at whether, for example, the maltose, the enzymes could be on the surface, they could be secreted, or they could be internal to the bacteria. And that could be helpful for understanding the structural mechanisms. Again, you could study this with light, and you could understand exactly where you'd want to try to apply a block to the bacteria, right? Do you want to get rid of the bacteria? Do you want to just block their product, et cetera, et cetera? So I think you've opened up a very good discussion about how you could attack these problems, and you've also connected it to a long-range health impact, which is that these byproducts actually have significant metabolic impact on people. And adding up all the sum total of all the different metabolic byproducts, having something that doesn't get digested as much could contribute to people not ending up with a long-term condition like diabetes. So I think being able to connect the basic grind of mechanistic understanding to long-term health impacts in a population is actually a very important product of science.
Okay. Sounds hard.
Any other questions from anyone? If not, then let's thank our speakers.